In the previous lesson in this series, we learned about how cells operate, and we used the simple bacteria type of cell as our example. In this lesson, we'll focus in on DNA and its role in storing the information needed to build these proteins, as well as its capacity to be duplicated to permit reproduction. You'll recall that with the emergence of life on Earth, Luca then had the wherewithal to build proteins to perform the chemistry of life. And Luca had hit upon a way of storing the information for these useful proteins in a molecule called DNA. And furthermore, that DNA molecule could be duplicated so that Luca would have the capacity for reproduction. By duplicating its DNA and passing on one copy to one offspring and the other copy to the other offspring, its descendants would have the information necessary to build proteins. So we'll see then that it's DNA that allows for organisms to pass on useful information about how to build useful proteins. And sometimes that DNA duplication involves errors, mutations, and this will generate new kinds of living things in the future. And that's what gets us from the origin of life to the origin of living things that have new traits like intelligence. It all has to do with this molecule of DNA. It stores the recipes for building an organism, but those recipes can change, giving rise to new kinds of living things with new features. Now we learned that cells use the DNA information to make proteins. Let's focus on the right side of the diagram here, and we'll see how this process occurs. It's a complicated process, but here we see the DNA double helix, and a region of that DNA molecule has been opened up. So this would be the recipe to make a protein in the sequence of letters along one strand. But in order to make the protein, the cell first copies the DNA information into a molecule of messenger RNA. Now there happen to be some quirky differences between DNA and RNA. RNA has a U instead of a T, but nevertheless the cell has a set of spare RNA letters that it will use to make a complementary strand of messenger RNA. So here's the template strand of DNA, and notice the letters are matching according to the pairing rules. So if there's a G in DNA, there's a C in RNA. If there's a T in DNA, there's an A in RNA. If there's a C in DNA, there's a G in RNA. But look, if there's an A in DNA, the matching partner, the complementary base, is a U for RNA. But notice then that the sequence of DNA is being used to build a sequence of RNA letters. And if you confirm the sequence of this RNA strand right here is identical to the other strand of DNA, so we can say that the RNA is a copy of this strand of DNA with the exception of the U in place of the T. So the cell's going to make this temporary copy of the recipe for building a protein and send it on out to the ribosome, the protein factory. The ribosome is this large molecular machinery that kind of plods along 3 letters at a time, and each 3 letters of messenger RNA specifies one of the 20 amino acids. We're not going to get into the details of all the relevant players, but there are carrier molecules called transfer RNA that are bringing amino acids to the site of the ribosome. And as the ribosome moves down the messenger RNA molecule, amino acids are linked together according to the sequence of triplets in messenger RNA. So this is how cells are building proteins. The sequence of DNA letters is being converted into a sequence of amino acids, which is what a protein is, a chain of amino acids. Let's use an animation now to see the 2 steps in the process of building a protein. The first step is to make a messenger RNA copy of the DNA sequence. Here we see DNA, and it is going to be opened up by a team of proteins, 2 complementary strands of DNA, and the red strand here is messenger RNA. You'll notice that messenger RNA is built from 1 strand of DNA. It is complementary to this bottom strand, and therefore it has the identical sequence as the top strand with the exception of U in place of T. Now the messenger RNA is going to detach from the DNA and then hook up to a ribosome. And it's here, then, the second step, where the ribosome is going to read the messenger RNA, in a sense, 3 letters at a time. These carrier molecules are bringing the amino acids. The ribosome will link the amino acids together in the growing protein chain. Eventually, the last triplet will be a stop triplet, and that will signal the ribosome the protein is finished. The protein will detach, fold up, and go about the cell to do its job. Now, let's introduce the genetic code over here, or just review the code. This is a depiction of the code. Notice it's in the letters of messenger RNA. We have a U instead of a T, and it is the language for life on Earth. So all living things are using this system because Luca came up with this system. And if we were to use the code here to figure out what's the identity of this first amino acid, well, the first triplet in messenger RNA is AUG. We go to the A quadrant in the inner ring, and then the U, and then the G, and that's methionine. So this first amino acid is methionine. The next triplet, CCC, is going to be CCC will be proline. So this amino acid is the amino acid proline. And we'll do one more. We have GUU. GUU is valine. So this third one would be valine. So the ribosome is simply going to proceed down the messenger RNA, each triplet specifying a given amino acid, and then the ribosome will make a strong chemical bond here. That is a covalent bond, a sharing of electrons between the amino acids. When the ribosome gets to the end of the messenger RNA molecule, there is a triplet that codes for a stop. So UAG, let's go over here to the code. UAG is a stop triplet, and this would signify the end of the message. So the recipe stored in DNA has a termination. There is an end to the recipe, and that is reflected in the final triplet in the messenger RNA, a stop triplet. There are 3 stop triplets, so each recipe for a protein has a stop triplet at the end, and that instructs the ribosome that the recipe is completed, and it detaches from the messenger RNA, and then the protein goes and folds up and then starts performing its job. So each of these different proteins that perform different jobs, they have a recipe stored in the DNA molecule as a sequence of letters along one strand. But not only that, the DNA can be duplicated, and the cell will duplicate the DNA in order to reproduce. And here we'll look on the left here. We'll begin on the left. Here's how the cell duplicates its DNA. The 2 strands separate, and then the spare letters of DNA in the cytoplasm are going to find their matching partners according to the pairing rules, the A with T and the G with C. And then the DNA polymerase will come along and chemically bond the letters together, so stitch the backbone together, forming the new strand. And notice then, there will be 2 new strands, one on the bottom and one on the top. And so you'll have the duplication of one DNA molecule into 2 identical DNA molecules. Over here on the right, we see that the bacterial chromosome is a circular chromosome, and so we've drawn here the beginning of the duplication process. Again, DNA polymerase will link the DNA letters together to form the new strand up top here and the new strand on the bottom. And that means we're going to have identical DNA strands, and the duplication will continue all the way around each of the circular chromosomes. Now, occasionally, mutations occur, and this is when there is a mismatch. So, for example, we see up here a T has been paired with a G, and that's not how it's supposed to happen. Also down here on this strand here, a C has been paired with an A, and that violates the pairing rule. So we would think of this as sort of a mistake. And that is what we term this, or we describe that as a mutation. And what that means is that you're changing the sequence of the DNA letters. And if this is the recipe for building a protein, that can affect the amino acid at that location and possibly the shape and function of a protein. So mutations produce new protein recipes, and this gives organisms the ability to change over time, and we call that evolution. Let's just confirm that a mutation can indeed alter a protein. Here's a messenger RNA sequence here with certain amino acids specified by the triplets. Let's say that we make a change in the first position of this triplet here. So we go from a GCG to an ACG. Well, a GCG would be GCG, that's alanine, so this triplet codes for alanine, so that amino acid is alanine, but an ACG is threonine. So a mutation here that changes the letter from a G to an A changes the amino acid from an alanine to a threonine, and that might affect how the protein folds up, and if it folds up differently, it might affect the function of that protein. It might lose its function, or perhaps gain a new function. So mutations change DNA information, which can change the shape of a protein, which can change the function, and therefore the cell as a whole. As mutations occur, then the offspring of the parent will be different from the parent and possibly different from each other. So we've seen that all cells have a boundary, the cell membrane. All cells have a fluid interior, the cytoplasm where the chemistry of life occurs. All cells are getting energy and resources from their environment. All cells have protein parts. These are the parts with purposes. These are little molecular machines that are doing different jobs, the chemistry of life. And now we've learned that these protein parts have recipes stored in DNA. So DNA is an information molecule that can be duplicated, and that permits the cell to reproduce.